In this theory video, we will discuss about inflammation. So, what is inflammation? Inflammation is a response of vascularized tissues to infections and damaged tissues that brings cells and molecules of host defense from the circulation to the site where they are needed in order to eliminate the offending agents. Complex local response of the living vascularized tissues to injury and mainly consists of responses of the blood vessels and the leucocytes, mainly neutrophils. So, the proper definition of inflammation is that inflammation is defined as the local response of living mammalian tissues to injury due to any agent. It is a body's defense reaction in order to eliminate or limit the spread of injurious agents, followed by the removal of the necrosed cells and tissues. Now coming to the agents that causes inflammation are, first we have infective agents. It includes bacteria, viruses, endotoxins, fungi, and parasites. Secondly, we have immunological agents that includes cell-mediated and antigen antibody reactions. Third, we have physical agents. It includes like heat, extreme heat, cold, radiation, mechanical trauma, etc. Fourth, we have chemical agents that includes like organic as well as inorganic poisons. Fourthly, and then lastly, we have inert materials such as foreign bodies. So, inflammation is distinct from infection. Inflammation is a protective response by the body to variety of etiological agents, both infectious and non-infectious, whereas infection is invasion into the body by harmful microbes and the resultant ill effects by toxins. Now coming to the signs of inflammation, the famous Roman writer named Celsus in the 1st century AD named the famous 4 cardinal signs of inflammations. First, we have the rubor, that is redness, tumor, that is swelling, calor, heat, and dolor, pain. Later on, a 5th sign called the functiolacia, that is loss of function, was later added by Virchow. Now, the word inflammation means burning. So, this nomenclature had its origin in the old times, but now we know that burning is only a sign of inflammation. Now coming to the types of inflammation, we have 2 types. So depending upon the defense capacity of the host and the duration of the response, inflammation can be classified as acute and chronic. First is the acute inflammation, which is of short duration that lasts less than 2 weeks and represents the early body reaction, usually resolves quickly and is usually followed by healing. Second, we have the chronic inflammation, which is of longer duration and occurs either after the causative agent of acute inflammation persists for a long time, or the stimulus is such that it induces chronic inflammation from the beginning. And the variant and another form called chronic active inflammation. This is also a type of chronic inflammation in which during the course of disease there are acute exacerbations of activity. So let's go into detail with one by one. First, starting with the acute inflammation. Acute inflammation is a type of inflammation which presents with all cardinal signs of inflammation within a short period of time or days, and it is associated with neutrophils infiltration. Now coming to the clinical presentations of acute inflammation, first we have the serous inflammation. This is a type of inflammation which inflammation with protein poor fluid, for example skin blisters that results from burning. Second, we have suppurative or purulent inflammation. This is a type of biogenic inflammation and pus formation in the inflamed area. Third, we have ulcerative inflammation that includes ulcer formation, loss of continuity of epithelium in the skin or mucous membrane due to inflammatory tissue damage. For example, is ulcerative colitis or peptic ulcer. Fourth, we have boils and furuncles. It includes focused skin collection of abscess, commonly in hair follicles. And last, we have pseudomembranous inflammation that includes like Clostridia, diphtheria infection, and characterized by formation of pseudomembrane composed of necrotic tissue and coagulated fibrin with neutrophil, macrophage, and bacteria. So, that are the clinical presentations of acute inflammation. Now, acute inflammation has two major components, namely reactions of blood vessels, that is vascular changes, and reactions of leukocyte, that is cellular changes. Now coming to the cellular events, it includes the alteration in the microvasculature, that is the arterioles, capillaries, and venules. It is the earliest response to tissue injury, and these alterations include hemodynamic changes and changes in vascular permeability. Coming to hemodynamic changes, this is the earliest feature of inflammatory response that results from the changes in the vascular flow and the calibre of small blood vessels in the injured tissue. And the sequence of changes includes: first is, irrespective of the type of injury, immediate vascular response is of transient vasoconstriction of arterioles with mild form of injury, and the blood flow may be reestablished in 3 to 5 seconds, while with more severe injury, the vasoconstriction may last for 5 minutes. Second is persistent progressive vasodilation, which involves mainly the arterioles, but to a lesser extent affects other components of the microcirculation like venules and capillaries. And the change is obvious within half an hour of injury, and vasodilation results in increased blood volume in microvascular bed of the area, which is responsible for redness and warmth at the site of acute inflammation. Third, we have the progressive vasodilation. In turn, may elevate the local hydrostatic pressure that results in the transudation of fluids into the extracellular space. This is responsible for swelling at the local site of acute inflammation. And fourth, we have the slowing or stasis of microcirculation follows, which causes increased concentration of red cells and therefore raises blood viscosity. Fifth, we have stasis or slowing, which is followed by leukocyte margination or peripheral orientation of leucocytes, mainly the neutrophils, along the vascular endothelium, and the leucocytes stick to the vascular endothelium briefly and then move and migrate through the gaps between the endothelial cells in the extravascular space. This process is known as emigration. So, what are the changes that we see in the vascular permeability? The pathogenesis includes, like, in and around the inflamed tissue, there is accumulation of edema fluid in the interstitial compartment, which comes from blood plasma by its escape through the endothelial wall of peripheral vascular bed. In the initial stage, the escape of fluid is due to vasodilation and consequent elevation of hydrostatic pressure. This is transudate in nature. But subsequently and gradually, the characteristic inflammatory edema exudate appears by increased vascular permeability of microcirculation. Now let's look into the mechanism of increased vascular permeability. In acute inflammation, normally non-permeable endothelial layer of microvasculature becomes leaky. First episode we have contraction of endothelial cells. The endothelial cells develop temporary gaps. In the later slide I'll show a diagram of all of this. So as of for now, please listen carefully. Contraction of endothelial cells. The endothelial cells develop temporary gaps between them due to their contraction, resulting in vascular leakiness. It is mediated by the release of histamine, bradykinin, and other chemical mediators, and the response is of short duration, usually 15 to 20 minutes. Now coming to the second, that is retraction of endothelial cells. Direct injury to the endothelium causes cell necrosis. Retraction of endothelial cells. We have in this mechanism there is structural reorganization of the cytoskeleton of endothelial cells that causes reversible retraction at the intercellular junctions, and the onset of response takes for about 4 to 6 hours after injury and lasts for 2 to 4 hours or more. Third, we have direct injury to the endothelial cells. Direct injury to the endothelium causes cell necrosis and appearance of physical gaps at the site of detached endothelial cells. And the increased permeability may either appear immediately after injury and then last for several hours or days, or may occur after a delay of 2 to 12 hours and last for hours or days, that is delayed prolonged leakage. And then fourth, we have endothelial injury mediated by leukocytes. So here, adherence of leukocytes to the endothelium at the site of inflammation may result in activation of leucocytes. Deactivated leukocytes release proteolytic enzymes and then toxic oxygen species, which may cause endothelial injury and increased vascular leakiness. This form of increased vascular leakiness affects mostly the venous, and it is a late response. Fifth, lastly, we have the leakiness in neovascularization. So in addition, the newly formed capillaries under the influence of vascular endothelial growth factor, that is VEGF, during the process of repair and in tumor are excessively leaky. Coming here is a picture of acute inflammation. Here we have the vascular end and the arteriolar end. So first, according to the sequence, first we have the gap due to contraction of endothelial cells. Second, we have retraction of endothelial cells. Third, we have direct injury. Fourth, endothelial injury by the polymorphonuclear cells, neutrophils. And then fifth, we have the new vascularization. So this diagram explains the whole of this. So I hope you have got the diagram. Now let's discuss about the cellular events. It has the cellular phase of inflammation has two processes. First is the exudation of leukocytes and phagocytosis. Let's look into one by one. First is the exudation of leukocytes. Number one is the change in the formed elements of blood. So there is slowing or stasis of bloodstream. With stasis, changes in the normal axial flow of the blood in the microcirculation takes place. Due to slowing and stasis, the central stream of cells widens and the peripheral plasma zone becomes narrow because of the loss of plasma by exudation. This phenomenon is known as margination. And as a result of this redistribution, the neutrophils of the central column comes close to the vessel wall. This is known as Wavementing. Second, we have the rolling and the adhesion. So peripherally marginated and wavemented neutrophils slowly roll over the endothelial cells lining the vessel wall. That is the rolling phase. This is followed by the transient bond between the leukocytes and the endothelial cells become firmer. That is the adhesion phase. Third, we have the immigration. So after sticking of neutrophils to endothelium, the former moves along the endothelial surface till a suitable site between the endothelial cells is found where the neutrophils throw out cytoplasmic pseudopods, and the neutrophils lodged between the endothelial cells and the basement membrane cross the basement membrane by damaging it locally with secreted collagenases and then escape out of the extravascular space. This is known as emigration. And the damaged basement membrane is repaired almost immediately. Fourth, we have the chemotaxis. The chemotactic factor-mediated transmigration of leucocytes after crossing several periods, that is the endothelium, basement membrane, perivascular, myofibroblast, and the matrix, to reach the interstitial tissues. That is called chemotaxis. First, this is a diagram of explaining the cellular events. First is the normal axial flow. Second is the migration and the wavementing. Third is the rolling and adhesion, where the neutrophils go and adhere to the vessel wall. And fourth is the immigration and diabetesis. So the neutrophils immigrate because of the release of collagenases. Lastly, we have the phagocytosis. Phagocytosis is defined as the process of engulfment of solid particulate materials by the cells. We call it as cell eating. So the cells performing these functions are called phagocytes. And the two main types of phagocytic cells are polymorphonuclear neutrophils. In short, we call it as BMNS. And secondly, we have the circulating monocytes and fixed tissues. Mononuclear phagocytes, commonly called as the macrophages. So phagocytosis involves 3 steps. First is the recognition and attachment. So I'll show you a diagram here. This is the recognition. I want you to clearly look at this picture for a second, to take a pause and then look at the picture. First, we have the recognition and attachment initiated by the expression of surface receptors on the macrophages. So we have the Fc receptor and Ig opsonin, and then we have the C3p receptor initiated by the expression of surface receptors on macrophages. We have the Mannose receptor and scavenger receptor. Second is the engulfment. The opsonized particles bound to the surface of phagocyte is ready to be engulfed and forms a phagocytic vacuole. Forms bigger vacuole called the phagolysosome. So the phagocytic vacuole, and then is engulfed. We have the phagolysosome over here. And lastly, we have the killing and degradation. So the microorganisms, after being killed by antibacterial substances, are degraded by hydrolytic enzymes. So that's all about the acute inflammation. Let's talk about chronic inflammation. Coming to chronic inflammation is defined as prolonged process in which tissue destruction and inflammation occurs at the same time. I repeat, chronic inflammation is defined as prolonged process in which tissue destruction as well as inflammation occurs at the same time. Chronic inflammation may be caused by one of the 3 ways. First is the chronic inflammation following acute inflammation. So when the tissue destruction is extensive, or the bacteria survive and then persist in small numbers at the site of acute inflammation, for example in cases of osteomyelitis, pneumonia, lung abscess, etc. That is chronic inflammation following acute inflammation. Secondly, we have recurrent attacks of acute inflammation. So when repeated bouts of acute inflammation culminate in the chronicity of the process, example in recurrent UTI infection, urinary tract infection leading to chronic pyelonephritis, repeated acute infection of the gallbladder that leads to chronic cholecystitis, etc. includes recurrent attack of acute inflammation. And third, we have chronic inflammation starting de novo. So when the infection with organisms of low pathogenicity is chronic from the beginning, for example like microtuberculosis, that includes chronic inflammation starting de novo. Now let's discuss about the types of chronic inflammation. So conventionally, chronic inflammation is subdivided into two types. We have non-specific and specific. Non-specific, when the irritant substance produces a non-specific chronic inflammatory reaction with the formation of granulation tissue and healing by fibrosis. Example is a chronic osteomyelitis, chronic ulcer, etc. Second, we have specific. Specific when the injurious agent causes a characteristic histological tissue response. Example, tuberculosis, leprosy, syphilis, etc. These are specific. And there is also a term called granulomatous inflammation. So granuloma is defined as a circumcised tiny lesions about 1 mm in diameter that is composed mainly of a collection of modified macrophages called epithelioid cells, and then rimmed at the periphery by lymphoid cells. And the word granuloma is derived from granule, meaning circumcised granule-like lesions, and oma, which is a suffix commonly used for true tumors, but here it indicates a localized inflammatory mass or a collection of macrophages. So a chronic inflammation in which epithelioid cells accumulate in clusters with central necrosis and giant cells surrounded by lymphocytes, plasma cells, fibrous tissue in a peripheral zone. This is called a granuloma. And inflammatory cells forming granuloma includes macrophages, plasma cells, and lymphocytes. For example is chronic granulomatous inflammation that includes tuberculosis, presents with granuloma containing Langerhans giant cells, central caseous necrosis, and other causes of granulomatous disease that includes leprosy, syphilis, sarcoidosis, and foreign bodies. So there is all about chronic inflammation. Now coming to the summary, we have discussed with acute and chronic inflammation. So let's find out the difference between these two. First, we have the feature here with acute inflammation, and we have chronic inflammation over here. So duration is for acute inflammation days or weeks. For chronic, it's months or years. And the types of cells includes in acute inflammation is mainly neutrophils. In chronic inflammation, it is mainly lymphocytes and the plasma cells, macrophages, and monocytes. And the cardinal signs all are present. And in chronic inflammation, some of them are included. And the healing process in acute inflammation is complete resolution, but in chronic inflammation, healing process is by formation of fibrosis. So inflammation is defined as the local response of living mammalian tissues to injury due to any agent. It is a body defense reaction in order to eliminate or limit the spread of injurious agents, followed by removal of the necrosed cells and tissues. Acute inflammation is of short duration and represents the early body reaction, resolves quickly and is usually followed by healing. While in chronic inflammation, we have longer duration and occurs either after the causative agent of acute inflammation persists for a long time, or the stimulus is such that it induces chronic inflammation from the beginning. So that's all about inflammation in brief. I hope you have understood. Thank you.